had the absolute pleasure of helping coordinate the High School Youth Researchers Summer Program this year, or HYRS. HYRS is a 6-week consecutive program that runs at the University of Alberta, University of Calgary, as well as the University of Lethbridge. The 8 incredible HYRS students have spent their summer working with various different research groups at the University of Lethbridge and gaining firsthand laboratory research experience. Tonight's showcase is really just a celebration of all of their work and their contributions to the biomedical and health research in Southern Alberta. There have been various individuals who have helped make the HYRS program successful, and I want to take a moment and really acknowledge all of their contributions. I would like to start by thanking Alberta Innovates for providing financial support for HYRS. Alberta Innovates plays a very pivotal role in establishing and promoting outreach programs that promote innovative health research. I would also like to thank the University of Lethbridge, the Office of Research Services, as well as all of the faculty and staff that have been involved in the HYRS program, especially Dr. Robert Sutherland and his team, Dr. Trushar Patel and his team, Dr. Stacy Wetmore and her team, Dr. Boris Demler and his team, Tony Montina and his team, as well as Dr. Uta Kote and her team. I would also like to extend a huge thank you to all of the postdoctorate, graduate, and undergraduate students of the University of Lethbridge who have supervised all the HYRS students this year. There are too many to name, but we are so grateful for all of your help, and we sincerely appreciate the time and the effort you have put in to make the experience of all the HYRS students truly enriching and positive. I would also like to extend a thank you to the Director of Youth Outreach, Valerie Archibald, for all of her support throughout the HYRS program. Valerie plays a very important role in our university community and gets more and more kids and youth involved and excited about STEM through programs like Destination Exploration. I would now also like to extend a thank you to Catherine Reeder. Catherine is our Director of Marketing and Communications for the Faculty of Arts and Sciences. Catherine has helped put together our program for the evening, and we are very grateful for all our help. Thank you all for making the HYRS program a great success. And yeah, now we will be hearing from some of our speakers for the evening. And I would like to start by introducing Dr. Janine Nguyen, who is the Associate Dean for the Faculty of Arts and Sciences, and I would like to invite her up to share a few words with us.
Thank you so much for inviting me. Oki, and welcome to the University of Lethbridge's annual HYRS Showcase. My name is Dr. Janae Nugent. I am the acting dean of the Faculty of Arts and Science. Usually I'm an associate dean, but I'm bringing greetings on behalf of the dean as he's away right now. I have the pleasure of welcoming you to this exciting showcase of the work of 8 highly intelligent and industrious high school students who have been working diligently all summer on some really exciting projects. Alberta Innovate sponsors this program as they recognize that these students, together with their incredible undergraduate and graduate student mentors, postdoctoral fellows, and faculty members of the University of Lethbridge, that they are forming the next generation of researchers. Oh, my light went off. There we go. With the challenges of global climate change and a pandemic, the importance of developing our talented youth so that they can become our scientific leaders of tomorrow is abundantly clear. The University of Lethbridge is very pleased to be able to support this Alberta Innovates program. Our impressive new state-of-the-art Science Commons building has facilitated collaborative and transdisciplinary research as these students have shared research space and lab materials with each other, with the master's and PhD students and supervisors, our talented pool of postdoctoral fellows, and with our world-renowned science faculty. I'm excited that they're able to share their projects with us all this evening. Congratulations to you all on such a successful summer of work.
Thank you, Janae. That was really nice. Thank you. I would now like to invite the Director of Youth Outreach for the Faculty of Arts and Sciences, Valerie Archibald, to share a few words with us.
Thank you, Zainab, for coordinating this wonderful event and the opportunity to showcase the projects the HYRS students have been working on over the past 6 weeks. I'm very excited to be here and to learn more about the work the students have undertaken. It's also exciting to see their faces without masks on and to learn who they really are. This group of 2021 HYRS students are the best and the brightest grade 11, soon to be grade 12 students in Southern Alberta. These students excel in academics and extracurricular activities. They balance work, school, and fun to be very successful young adults. This summer, the University of Lethbridge had 8 students join various STEM labs on campus to experience hands-on research and learning opportunities. The students have been assisting with current research and learning new lab techniques. They've had the opportunity to join current research teams and personally experience the work happening in these labs. In the past, we've seen a small number of women and BIPOC in STEM. This number is slowly increasing, with Statistics Canada reporting that 23% of science and tech workers were women in 2016. This evening, you will see a group of diverse students representing many cultures and 75% identifying female. This is great news for the future of representation in STEM moving forward. This is also very exciting for the University of Lethbridge, as it reflects the commitment of the institution to expanding equity, diversity, and inclusion on campus and within the community. The University of Lethbridge supports our young learners by providing space, programs, and opportunities for those as young as 5 to be a part of the learning community on campus. The new Science Commons building houses a state-of-the-art outreach facility that gives the opportunity for school-age learners to come to campus and to participate in STEM-based activities and learn more about research and campus life. The University of Lethbridge is also home to the award-winning Highschool iGEM team. When young students join the university community to learn STEM, participate in programs, and dive into the research work that is currently taking place on campus, they start to build a sense of belonging. Over time, the University of Lethbridge becomes their university and a part of their academic story. Through programs like Destination Exploration, Let's Talk Science, iGEM, and the HYRS program, thousands of young people have experienced the enjoyment of STEM at the University of Lethbridge. From joining the Destination Exploration team as a volunteer in middle school, to participating in the HYRS program in high school, and naturally transitioning to a University of Lethbridge student, and now the student coordinator of the HYRS program, we see how outreach, research, and the university community has played a role in the life of Zainab. Zainab has been the HYRS coordinator for the 2021 season and has risen to the challenge and exceeded all expectations for this role. She has provided learning and social opportunities for the students, created a safe space for them to ask questions, learn about campus, and gain new skills over the summer. I've thoroughly enjoyed working alongside Zainab this summer and owe her a huge thank you for her tireless work and making the HYRS program a fantastic success. Thank you, and I look forward to seeing the presentations this evening.
Thank you, Valerie, for your very kind words. I would also like to thank you for all of your support and encouragement throughout the program. It's been an absolute pleasure working alongside with you. Now I would like to invite Trinity Deek. Trinity is one of the alumni of the HYRS program, but also one of her undergraduate lab supervisors, to share a few words with us.
Hi, my name is Trinity Deek, and I'm an alumni from the HYRS program from 2018. During my time in HYRS, I got to work in Dr. Stacy's Wetmore's lab doing computational chemistry. This was a really amazing opportunity for me to get hands-on experience in a field of chemistry I had never even heard of before the program started. This program was my first introduction into research and how all of it was conducted. My lab provided a phenomenal learning environment where I learned lots about my project alongside with information that helped a ton in my first year bio and chem classes. Overall, the experience of HYRS has taught me so much about research, various science fields, and university life in general. Between all the tours, guest lectures, and hands-on research in my own lab and the labs of my HYRS peers, I really got the chance to understand all of the complexities and opportunities research had to offer. I never realized before the program that doing research could be a career option, or that it was not only in institutions like universities, but also government and industry. It's opened my eyes to lots of other jobs and career paths that I didn't know existed. Walking into the program, I had no idea I'd be walking out with some of my best friends and a lab I loved being a part of. HYRS has given me lots of opportunities since starting university. These include research opportunities and grants, along with early connections with professors and researchers here at the U of L. Working in a university lab is a pretty competitive job once you start in university. My HYRS background has made this a lot more accessible. This year, I was also able to become a mentor for the program, as Zainab said, and I've been able to mentor Zaki, a current HYRS student. It was really cool to have HYRS be my first introduction into both research and high-level chemistry, and then be able to turn around and be part of someone else's first exposure. During the program, from both the student and the supervisor, has made me appreciate all this program can offer to high school students. I'm very grateful for both chances I had to be involved in the program, and I hope that you all as students have enjoyed your experiences just as much as me. Thank you all for letting me speak, and I look forward to hearing what all of you guys have been working on over the summer.
Thank you, Trinity. That was great. And I would like to thank you for being actively involved and really mentoring the next generation of researchers. Now I would like to invite Finley Lothian. Finley is one of our wonderful HYRS students to come share a few words on behalf of the HYRS cohort.
All right, good evening, everyone. My name is Finley Lothian, and I've had the wonderful opportunity to speak on behalf of my fellow HYRS group members this year. So we've all had the privilege of working within various labs, notably the Cote, Wetmore, Demler, Patel, Montina, and Sutherland labs. So throughout our varied experiences, we've all been able to learn, grow, and explore, working side by side with incredible lab mates and esteemed professors. The program has provided us with both nurturing and collaborative environments that have allowed us to explore without the fear of failure. Our HYRS group cannot say enough about the relationship and the mentorship that have occurred over the past 6 weeks. Everyone has been so willing to lend a helping hand, while we have been able to uncover the many areas of interest that are possible in the next chapter of our lives. I would be remiss if I did not take the moment to note that all of this would not be possible without the help and support of 2 key members of the HYRS program at the University of Lethbridge here, Miss Valerie Archibald and Miss Zainab Enatuela. Thank you all for what you have done for us the past 6 weeks. It has been so appreciated. Words cannot explain. And finally, we would collectively like to thank Alberta Innovates for providing Southern Alberta students with this tremendous opportunity. This HYRS experience has truly gone beyond the school textbooks that we read and learn, and brings to light what is possible within the world of STEM. I can confidently say that I am excited to see what lays ahead for my HYRS group members, and if this summer is any sign of the future, we are in for some inspiring journeys.
Thank you, Finley. You are very kind as always. We will now be moving on to the presentations, but before that, I would really like to take a moment to acknowledge all of the HYRS students for all the hard work that they have put in the past 6 weeks. The foundation of the HYRS program are the incredible students that participate every year. I have had the absolute pleasure of getting to know each and every one of them over the summer, and I can confidently say that this is one of the brightest and most hardworking group of people that I have ever met. I'm incredibly proud of each and every one of them, and as is their university community. A couple housekeeping things just before we get started on the presentations. Please keep yourself muted during the presentation. And after each presentation, you will have an opportunity to ask any questions that you have. To ask your questions, you can either leave it in the chat and we can read them out. If you're putting it in the chat, just make sure that you put the student's name, who the question is for. You could also raise your hand and ask your question that way, if that's easier. For each presentation and Q&A session, we have 10 minutes allotted, and we will be moving on after the 10 minutes. So if you have a question that has not been answered during that time, we will have plenty of time towards the end after all the presentations are concluded. So you can have an opportunity to ask your questions then, and I'm sure our students will be very happy to answer them. Yeah, and then like I said before, you can click view on the top right of your screen and select speaker, side-by-side speaker view, sorry, and then you can witness both the presentation as well as our speaker simultaneously. Yeah, the session is being recorded, just as a reminder for anybody who would like to turn off their cameras. But yeah, with all of that being said, I would like to introduce our first presenter for the evening, Jorja Salmon. Jorja is representing Raymond High School in the HYRS program, and over the summer, she has worked with Dr. Robert Sutherland and his team in the Canadian Centre for Behavioral Neuroscience. I have not spent 6 weeks on these projects. I'm not going to attempt to describe them, and I would like to invite Jorja to share her work with us.
Hey, everybody. Like Zainab said, my name's Jorja. I was part of the Southern Lab this summer, which is a great lab. And my project was on the area of the hippocampus within a mouse model of Alzheimer's disease. So a little bit of background, Alzheimer's disease is a buildup of amyloid plaques and neurofibrillar tangles within the brain. Amyloid plaques are misfolded proteins that form in the spaces between neurons, and neurofibrillar tangles are abnormal accumulations of a protein called tau that also gets collected inside the neurons. So when your brain gets Alzheimer's disease, you start to lose control of some functions, like you can lose some memory, and sometimes it can even inhibit your ability to hold conversations. So for my research project, we were focusing on an area of the brain called the hippocampus, which plays a major role in memory and spatial memory and things like that. And so specifically in this project, we were measuring the volume of the hippocampus when it has Alzheimer's disease at various ages. So to start off our project, we had 24 female mice. Half of them were used as a control group to compare, and then the other half we had at different ages. So some of them were 3 months, some of them were 6 months, 9 months, and 12 months. So you got to see the disease throughout like different ages, kind of gave us a real idea of it. And so we perfused the brains, and then we stained them so we could see the formation better. And then we looked and manually measured the area that the hippocampus was through this program. And so in the bottom thing, sorry, it's not letting me zoom in, but in the bottom corner, the 6 months negative picture versus the 6 months positive picture shows you kind of what pictures we were looking at and what the results were. So then we would go and measure the hippocampus in those, which is like that little dark spot has like a darker area in it. We would go through and measure that, and then compare it to the ones that did have Alzheimer's disease and the ones that didn't. And so up here in results, we have the orange graph, and that one shows you like the area of the hippocampus at different ages. So the first one's 3 months, and it goes all the way to 12 months. Kind of shows you that as the mice do get older, the hippocampus area does get smaller when they have the disease. And then our blue graph over on the other side shows you roughly what the difference is between the ones that do have Alzheimer's disease and the ones that don't. So the one that's smaller over there, the 3 months positive, is the one that does have a model of Alzheimer's disease in it. And when you compare it to our control group, you can see that it does have less area in it. So in conclusion, just to kind of sum it up for you guys, we looked at the hippocampus in these 24 mice and then compared the ones that did have a model of Alzheimer's disease to the ones that didn't. And Our hypothesis of the ones that did not have Alzheimer's disease having a bigger hippocampus was confirmed. And yeah, these are results. I'd like to acknowledge the HYRS program for letting me be a part of it this year, and a huge thanks to the Sutherland Lab for letting me join their lab this summer. It was really fun to tag along and see all their research projects.
Thank you, Jorja. That was amazing. We will now be taking some questions. So if you have some questions, you can leave it in the chat, or again, use your raise your hand feature. But to get us started, I have a question. For your graph in the blue, is the difference in the hippocampus going to be the same at the 12-month mark? Because that one is for the 3-month mark, correct?
Yeah, so I think that in the 12-month thing, there would still be a difference, obviously, but I think the difference would be greater, just because as the disease goes along, I feel like the area of the hippocampus would be less. But yeah, so the graph would be slightly different. I was just giving you a general idea of the difference with that graph.
Thank you. There is a question for you in the chat, so I will read it out. What was the favorite activity that you've done in your lab?
Favorite activity would probably be when we got to watch the electrode surgery on a mouse. For the electrodes, they stick them in the brain, and then it measures like the electric energy, so you kind of get an idea of like what's happening and where the most activity is happening in the brain. And we got to watch that get put in, and that was probably the best experience I had in the lab.
That does sound pretty amazing. Yeah, another question, sorry, but for what would be the future directions of your research here?
So I think that this research could just be used as a baseline for other research. So because we know now that hippocampus is smaller in mice that do have a model of Alzheimer's disease, we could take it and we could use it to like test further, like, all right, because each individual mouse varies, right? Because no individuals are the same. So you could test like, okay, this one has a smaller hippocampus than this one does, and they're both 6 months. So it could be like, what one's experiencing more severe symptoms? So things like that. I think this would just be a baseline for further research.
Great, thank you so much. That was an amazing presentation. Thank you, Jorja. We will now be moving on to our next presenter. Our next presenter is Priyanshi Patel. Priyanshi is representing LCI in the HYRS program this year. Priyanshi, alongside Jorja, has also worked with Dr. Robert Sutherland and his team in the Canadian Centre for Behavioral Neuroscience. And I would like to invite Priyanshi to share her research with us.
Hi, good evening, everyone. So my research was based on using fear conditioning to analyze behavioral changes and cognitive impairment that occurs in an Alzheimer's disease mouse model. So just an introduction, Alzheimer's disease is a neurodegenerative disease and a form of dementia. Usually the pathologies that develop in the brain from Alzheimer's is amyloid beta proteins, which accumulate and form plaques in the brain, and then the tau protein, which forms tangles in the brain. Due to these pathologies developing in the brain, usually the neurons, their ability to communicate for metabolism, repair everything tends to get disrupted. The technique I focused on is fear conditioning, which is an associative learning task where the mouse is presented with a tone followed by a mild foot shock. And the idea is that they will eventually adapt a freezing behavior, either just by being in the context that they were shocked in or hearing the tone. So in the fear conditioning task, it occurred over a duration of 4 days. On the first day, it was just training the mouse to the tone and the shock and allowing them to adapt the freezing behavior. On the second, third, and fourth day, we tested them we did different tests on them, like the context test, the neutral test, and the tone test. The one I focused on was the tone test, which is where you play a tone for about 20 seconds, and then that happens for a total of 3 separate tones. So the data we collected was for the table, it just shows a normal fear conditioning data for Alzheimer's disease mouse model that has a fast generation of amyloid beta protein. The RP80 mouse model demonstrates significant freezing when it's at the 2.5-month-old mark. However, the freezing time does tend to decrease as you look at an older mouse model, such as at the 4-month mark. The graph is just comparing the 4 different groups of mice that were tested and just seeing and comparing their freezing percent at different ages. So at 1-month-old, 2.5-month-old, and then 4 months old. So the conclusion that we came up with was that the mice were initially able to learn that basic conditional paradigm, that they were able to associate the tone with the shock and exhibit that freezing behavior. These data just suggested that the mice initially did learn the tone and their memory was enhanced at the 2.5-month mark. However, that begins to drop at the 4-month-old age for the RP80 and the SL80 mouse models. Fear conditioning overall is has been proven to be a pretty effective technique and study for Alzheimer's disease mouse models, and just to have a better understanding of how they go through different behavioral changes and cognitive impairment over time. Before I end off, I would just like to acknowledge the HYRS program for giving me the opportunity to be a part of their program over the summer, and Dr. Sutherland for allowing me to be a part of his lab, and the entire lab team for being so supportive and providing great guidance throughout the entire 6 weeks. And I'm open for questions now.
Great, thank you, Priyanshi. That was great. Yeah, I will start off with the question for you. You suggested, I think your data suggested that the mice associated context and tone with shock. Was this result sort of expected, or were you surprised at all?
No, I'd say it's a pretty expected result, because it's just like training the mice to the whole tone shock pairing. So it's just the idea that over time, as they if you look at an older mouse model, that freezing time begins to decrease, and that has been noticed in a lot of these mice as well. So I would say it was a pretty expected result.
Great, thank you. I'm also going to read out a question that is in the chat for you. Were there any non-hypothesized effects of these similarity tests that was common on the multiple mice, if that was analyzed at all?
That is a good question. Is it okay if I come back to that one?
For sure. Do you want to take the next question for now?
Yeah, for sure.
What was your favorite thing that you've done in the lab, Priyanshi?
So honestly, everything we did was great because it brought a different learning opportunity in its own way. I would personally say, just like Jorja, the surgery was really cool to witness and just be there to see how the entire process works. And just the basic sectioning and mounting that we did with the mice brains, that was also a really fun experience.
Great, thank you so much. Yeah, I have another question for you. What do you think are some of the future directions with the research that you have done this summer?
I would say maybe this is just a basic, I guess, research. And then one of things that could be done is maybe just looking at if the maturity of the plaques and tangles is something that would affect something like fear conditioning, rather than just the age of the mice. So as they grow older, that freezing time begins to decrease. Instead of just having that age aspect, there would maybe the maturity of the mice be something that affects it.
Yeah, for sure. Thank you. Yeah, if you want, we can come back to the question at the very end of the presentation if you would like.
I mean, yeah, sure. About the non-hypothesized one, I'm not sure if there was anything non-hypothesized that was like a result from the test. So.
Yeah, that's great. Thank you. Thank you, Priyanshi. I appreciate that. Yeah, we'll be moving on now to our next presenter. Our next presenter is Eva Glenn. Eva is representing Chinook High School in the HYRS program. He has worked in Tony Martinez Magnetic Resonance Lab in the Department of Chemistry and Biochemistry. I would like to invite Eva to share her work with us now.
Alrighty. Hi, everyone. As Zainab said, my name is Eva, and I've had the chance to work in Tony Martinez Lab, which is the Nuclear Magnetic Resonance Center this summer as a HYRS student. So I was coincidentally assigned to the same lab placement as Trinity Ramos, who will be presenting after me, who I also attend Chinook High School with. So Trinity and I did quite a bit of work together in the lab, and we both contributed to each other's projects. So my project is called An Introduction to Metabolomics and Nuclear Magnetic Resonance Spectroscopy. So I'm just going to zoom in really quick. Okay, so starting off with our introduction, when Trinity and I work in the lab together, we use a field of study called metabolomics, which is the large-scale study of tiny molecules called metabolites. Metabolites are all the downstream byproducts of cell metabolism, which is a process that cells use to create energy. These metabolites can be anything from sugars to proteins to fats and things like that. Cell metabolism allows organisms to grow and reproduce, maintain their structures, and respond to environmental changes. In the human body specifically, we can study these metabolites by looking at human cells, tissues, or biological fluids. This can be things like blood, urine, brain tissue, or things like that. So when we're analyzing these metabolites, we're going to need an instrument that goes to the molecular level to tell us which metabolites are present in the sample. So this instrument is known as the Nuclear Magnetic Resonance Spectrometer, and abbreviated is NMR. So I'm sure most of you have heard of an MRI machine before. The NMR is basically just a scaled down version of an MRI. So this photo on the right in our introduction here is what the NMR looks like. It's like an MRI, but it's smaller and it's placed vertically, so the samples go in on the top. It uses similar principles from an MRI to scan samples for specific metabolites. It creates a big magnetic field that when a sample is placed inside, these signals it creates within the machine produces its own fingerprint. And we can analyze this to tell us exactly which metabolites are present. So moving on to methods. When we're scanning a sample in the NMR spectrometer, we're going to need to do some preparation on the sample first. Each kind of tissue or biofluid has a specific method of preparation. During our time in the lab, we worked with urine and blood and prepared them to be run in the NMR. So this chart, figure 1 with the 3 boxes here, you can see the different methods of preparation. So we use 2 different methods on blood and 1 on urine. I'm not going to go into too much detail about the exact steps that we took, but it's pretty clear to see that urine, for example, takes less steps than the methanol extraction method in blood. So after the sample prep, we can run the samples in the NMR. And when this is done, we will get a spectra that's made up of many different peaks that we are able to analyze. Using statistical tests, we can create data sets that will help us compare our results. We then do what's called metabolite identification, where we go through and fit our spectra with a specific curve that represents a possible metabolite. Once we identify all significant metabolites, we do what's called pathway analysis, where we take the identified metabolites and put them into a software that shows us the pathway in the body that incorporates that metabolite. We then can apply this in a clinical setting, obviously depending on the experiment, and our results will be helpful for further research and diagnostic techniques. So moving on to our results. As you can see in this figure on the top here, this is what the spectra looks like once it's run through the NMR. So each of these individual peaks represents a specific metabolite. So this particular spectra was obtained from when we ran our urine samples in the lab. Next, we do a process called binning, which is just splitting up every single peak in the spectra. So as you can see, the photo on the left with the green and red lines that split it up, marking the beginning and end of each peak. Once we do that, the computer software tells us which metabolites are statistically significant, and we can go through and identify them, which is what the figure on the right shows. This list at the bottom of the photo shows any possible metabolites that the software thinks could fit these peaks. We then go through this list, and it'll give you a shape to match up to the peaks. Obviously, there will be some curves that look nothing like the peaks, but in this photo, you can see that the blue area under the curve fits perfectly with this specific peak. So after we do that, we get a set of data, that looks like these 2 graphs. This is done through statistical analysis. This is a data set collected from Mike's project, who is one of the grad students we got a chance to work with. His study was done in collaboration with NASA to assess possible long-term health impacts of spaceflight. On the left, this graph is taken from blood samples, and on the right is from urine samples taken from the same astronauts at the same time points with the same diet and other similar conditions. The red squares represent the samples taken from pre-flight, and the blue triangles were taken on flight day 15 in space. The x-axis represents the separation between the blue and red groups, and the y-axis is the separation within the groups. So you obviously don't need to be able to read this graph too well to tell me that there is clear separation in the urine graph, but there is overlap in the blood graph. This graph will tell us that using different biofluids can tell us very different things about our experiment. So in this situation, urine can tell us much more about the changes in the metabolome. So we then do what's called pathway analysis. So once we are confident in our metabolite identification, we put it into a website called Metabolomics, and it shows us the pathway of that molecule in the body and how it's altered. These are pictures on the top left and right here of what the website gives you. So both are similar diagrams, just in different software. So this is a pathway analysis we did of our urine samples that shows the metabolism of ketone bodies. The website can also create a graph showing us which pathways are most significantly altered during our experiment. So you can see on the bottom right here. The higher on both axes a circle is, the more impact it has on the pathway. So the ketone body metabolism is that big red circle in the top right corner. So obviously it is shown it has a very big pathway impact. So lastly, on to our conclusions and future directions. So some of the biggest takeaways from using metabolomics and NMR spectroscopy is how applicable it is to other studies. Metabolomics can help in things like pharmaceutical interventions, diagnosing disease, and observing metabolome alterations from external conditions like drug use or diet, for example. NMR spectroscopy is very helpful in identifying and quantifying significantly altered metabolites. This technology is very versatile in its applicability to almost any other field of scientific study. Okay, and so for references, I'd like to thank the following individuals and funding sources and the HYRS program and the Montina lab especially. And that's all I have, and I'm open to questions.
Thank you, Eva. It was an amazing presentation. Yeah, I, like always, have a question. This is about figure 2, I believe, that's on the left. As someone who's clearly not part of this field, what do the different colors on the graph actually indicate?
Yeah, so as I mentioned before, this spectra was obtained from when we ran the urine samples in the NMR. So the different colors are each individual urine sample. So we ran a ton of urine samples in our time in the lab, and this data set is just all the samples combined into one spectra, so it's easier to analyze.
Yeah, there's some great comments for you. We've been saying you did a great job, Eva. Another question for you. You said that this field is very applicable in various different scientific research. I was just wondering, what are some other areas where this would be applicable in specifically?
Yeah, so in the NMR lab, for example, we have a variety of different students working in all different majors, like for example, biochem or neuroscience. And this proves that many different fields of research benefit from metabolomics and can use the NMR in their studies. And NMR is also very applicable into the health sciences. And so when Trinity presents, she'll talk about some of the more applicable things. And so, yeah.
Great, thank you so much. Thank you, Eva. There is a question for you in the chat. What did you find challenging during your experience with HYRS?
I'd probably say learning NMR and metabolomics as a whole, because it's an entirely new and different field of study than what I've ever been introduced to. And so being able to learn different stuff that we don't usually learn in high school courses, I think was a really nice experience, but was also challenging.
You guys excelled at it. Thank you, that was a great presentation. We will be now moving on to our next presenter. Like Eva mentioned, our next presenter is Trinity Ramos. Trinity is also representing Chinook High School in the HYRS program this summer. Trinity and Eva both have worked in Tony Martinez's magnetic resonance lab in the Department of Chemistry and Biochemistry. So I will like to invite Trinity now to share her research with us.
Hi, everyone. So over the summer, like Zainab and Eva mentioned, I had the opportunity to be a HYRS student in Tony Martinez's lab, where Eva and I work together. And so this poster is sort of the second piece to our big HYRS project. So my poster and my project is called Proton NMR Analysis of Urine Collected from Varsity Athletes within different time periods of experiencing a concussion. So concussions are a type of traumatic brain injury, or TBI, and it's usually caused by the brain colliding with the skull's hard interior. So this disrupts normal brain functions, and chemical reactions in and controlled by the brain are altered. Concussions also increases the chances of brain swelling and disability, and in extreme cases may cause death. And this is more concerning because studies have shown that the risk of concussions is higher among youth, especially young athletes. Another concern is the subjectivity of the diagnosis. So without quantitative evidence, clinicians heavily rely on patient-reported symptoms, which makes it difficult to accurately gauge the damage caused and the recovery needed by each individual patient. So using data collected from varsity athletes of the University of Calgary, we set out to identify distinct changes in metabolites that correlate as a cause of the TBI. So as you can see on figure 1, the methods are very similar to the process that Eva already described for urine. So first we organize the samples according to individuals' baseline, days 1 to 5 post-concussion, which we labeled D1, D2, and so on, and return to play, or RTP. So the only other difference was that the urine was a bit more acidic. So in order to get it closer to a pH of 7.4, which is what's ideal for the NMR, we had to adjust the ratio of buffer and TSP to sample. Everything else is identical, so after running it in the NMR, we separated each peak of our spectra into bins. We ran statistical analysis to compare overall changes and to acquire bins that were significantly altered. From there, we identified these metabolites using a computer software called Genomics, and once we were confident with the metabolites we identified, we analyzed them based on relevance to our study and on their impact on specific metabolic pathways. So for results, we came up with 2 main comparisons. So baseline, which are control samples, versus day 1 post-concussion, which is the graph on the left. And then day 1 post-concussion versus return to play, which is the graph on the right. And using statistical analysis, we found that there is clear statistically significant separation between each group within each comparison. So on these figures, the x-axis shows the separation between the 2 groupings. So the circles show how the samples cluster together. And the y-axis shows the separation between the samples in each group. So the difference between this triangle versus this triangle and so on. Therefore, we can say with some confidence that the metabolites in each athlete were significantly altered from baseline to day 1 and from day 1 to return to play. Another analysis we did was the time series comparing metabolites over a span of 5 days post-concussion. So this analysis compares the concentrations of metabolites in specific bins, and the most interesting one that we found was the identification of picolinic acid. So according to our research, it's produced under inflammatory conditions, which could indicate the brain swelling caused by the concussion. Lastly, out of the significantly altered metabolites that we identified, we analyzed what metabolic pathways in our body they had the most impact on. So while the metabolites we found don't guarantee a direct correlation between these pathways to concussions, it's worth noting what roles they play in our body. So for figure 4, the graph on the left shows that the most significantly impacted pathway when comparing baseline to day 1 samples is the citrate cycle. And in other studies we looked at, adult male rats that sustained a TBI saw a decrease in glucose metabolism, which is a crucial stage to begin the citrate cycle. This is because it's redirected more towards energy production to help with the injury. Moving on, the graph on the right shows histidine metabolism to be the most significantly impacted pathway when we compare day 1 to return to play. So to understand the significance, other studies looked into how mast cells respond to traumatic brain injuries in concussed mice. And mast cells are important because when they recognize an attack, like from things like infections or pathogens, they release chemical granules, one of which is histamine, to help protect and heal the body. So histamine is an inflammatory molecule, which relates back to the significance of histidine metabolism and demonstrates its role in the inflammatory response, again, likely caused by the traumatic injury of a concussion. So to conclude, the current diagnostic tools for concussions can be subjective, which limits the accuracy of detection, therefore a safe return to play for athletes. There is clear separation in the altered metabolites when comparing baseline to day 1 samples and when comparing day 1 post-concussion to return to play samples. Picolinic acid was a significant metabolite in the 5 days post-concussion time series, which may have ties to inflammation during the onset of the TBI. And pathway analysis showed that the altered metabolites had significant impact on the citrate cycle when we compared baseline to day 1, and on histidine metabolism when we compared day 1 to RTP, return to play. Our future directions involve an improvement of diagnostic tools and the identification of biomarkers. So this means a better and quicker understanding of the damage caused by a concussion and the best treatment needed. We also would like a more robust sample size to consider other factors like varying lifestyles, body composition, age, relation to past injuries, and possibly a multi-concussion study that could identify differences when looking at the return of metabolites to their original state after recovering from a concussion. So we'd like to thank the following people, and here are my list of references. Thanks for listening, everyone.
Thank you, Trinity. It was a wonderful presentation. You did a great job. Again, question for you. What was the sample size for your experiment?
So our sample size, we're actually waiting for more samples from the University of Calgary at the moment, but out of around 270 urine samples, we narrowed it down to 6 individuals with enough data that we could run complete comparisons. So all 6 individuals had days 1 to 5 post-concussion. So we got to do the time series. And then only 2 had the complete baseline day 1 and RTP. Another 2 only had baseline and another 2 only had RTP. And we also wanted to make sure that we accommodated to make sure that it's within subject design, which just means that it's only valuable if we compare one individual's baseline to their own day 1 post-concussion and their own RTP samples.
That makes sense. Thank you, Trinity. Another follow-up question. I know for your future directions, you had mentioned just establishment of better diagnostic tools. Any suggestions on what some of this may be?
Yes, so we wanted to find biomarkers that we can identify. Sorry, that's my dog. So we wanted to see if we can get diagnostic tools like, say, a urine test or a blood test that we could run in the lab and easily see important information like how much damage or what degree of recovery that person needs. This would also be more reliable in case an athlete could downplay what symptoms they have, or when symptoms just don't show up the same way in everyone else.
For sure. Thank you, Trinity. That's a wonderful presentation. We will continue with our second set of presentations now. And our first presenter is Amber Quill. Amber is representing Winston Churchill High School in the HYRS program. Amber has spent her summer working with Dr. Tushar Patel and his team in the Department of Chemistry and Biochemistry. So now I would like to invite Amber to share her research with us.
Thank you, Zainab. So I had the honor of working in Dr. Tushar Patel's lab. What I worked on this summer was investigating flaviviral non-coding RNA interactions. So flaviviruses are, they have a positive strand viral RNA genome, and it consists of viruses like Zika virus, Japanese encephalitis virus, Powassan virus, and Murray Valley encephalitis virus. These are pretty dangerous because JEV, or Japanese encephalitis virus, is the leading cause of viral encephalitis in Asia, with about 68,000 clinical cases each year. What we investigated this summer was the 3' here and 5' non-coding terminal regions of the viruses. So in between is the coding areas, which codes for anything that the virus needs and codes for. And the 3' terminal region and 5' terminal regions are non-coding. So, so they don't code for anything, but they are still very important. What we have been looking at was the viral cyclization sequence and how that's basically how the 3' and 5' terminal regions interacted with each other. So our focus was primarily the 3' and 5' terminal regions, and also we experimented on the high bind, which is this green shaded area here and this red shaded area here. So that's just smaller portions that we ran our experiments on. It's theorized that it's proven that the cyclization sequence is involved with viral replication, and it's been theorized that they bind together. So we have been currently exploring in order to better understand the implication on replication of the terminal regions. Here is a computational prediction of the secondary structure. So here in the red is the 3' terminal region, and here in the green is the 5' terminal region. What we did in order to get our RNA was we transformed it, purified it, linearized it, in vitro transcribed it, and then had purified RNA through size exclusion chromatography. And we examined our interactions in solution by labeling it and then doing microscale thermophoresis, or MST, as well as doing SEC-MALS, which is size exclusion chromatography, multi-angle laser light scatter. In order to visualize our interactions, we did size exclusion chromatography, small angle X-ray scatter, in which we sent our results, we sent our RNA in to get it tested in Oxford. Here is our fluorescent labeling. This is the process that we followed, which is basically putting all the things we had in there, and then as well as the RNA. We centrifuged it. It would come to the bottom like the non-labeled RNA. We would add buffer and continue centrifuging it in order to purify it and have just our labeled RNA. And here is our chemical reaction diagram of what's going on while we're doing that. Through MST, or microscale thermophoresis, there's just a laser which shoots the laser through 16 capillary tubes. One concentration of our either 3' or 5' is remained constant throughout all of the 16 capillary tubes, and the other concentration is serially diluted, or basically cut in half each tube. And we do this in order to measure the difference of the fluorescent migration of a bound versus unbound target, which is, we use this in order to find the dissociation constant, or the KD. Here is our Japanese encephalitis virus, JEV high bind RNA. This is our size exclusion chromatography chromatogram. So here there are different fractions which we took of where there's high peaks here, means that there's RNA there. And so we took where the peaks are through the 5' here and the 3' here, and we ran them on a urea polyacrylamide gel electrophoresis, or urea page. And since we ran them and they're straight across, it means that basically that they are all the same size and that all our RNA is the correct size, which is about 220 nucleotides. So that means that it was successful. Up here is our Powassan virus terminal regions RNA. So here are the peaks again from the chromatogram. And we took the fractions, which are they are 0.5 milliliters or 500 microliters. We took the fractions here and we ran these on a urea page gel as well. And we got them at about 114 nucleotides and 108 nucleotides, which means that, again, since these are straight across, they're the right length, and we have successfully purified our RNA. Here is our MST diagram. As you can see, the curve here is a good curve. We found that our KD, or dissociation constant, was 59 plus or minus 9, or about 60. And what we did was we serially diluted the JEV3 prime, and the JEV5 prime remained constant. And as you can see here, it's a good curve. So what we did this summer was purified Japanese encephalitis virus, Murray Valley encephalitis virus, Powassan virus, both the three-strand terminal regions and the 5-strand terminal regions of all 3 RNAs. And through gel electrophoresis, we had a constant band at an appropriate nucleotide length. We also labeled our RNA of 1 part of each. And then through MST experiments and SEC-MALS, we've proven that the JEV 3-strand terminal region and 5-strand terminal region interact in solution. And then our future directions are we're going to look forward to performing MST experiments on Murray Valley encephalitis virus and Powassan virus in order to explore their cyclization, and as well how they interact in solution. I'd like to just say thank you to HYRS, U of L, as well as Dr. Trisha Patel, Tyler Misowich, and Sean Park and Corey Nelson for letting me work in their lab.
Thank you, Amber. That was a very nice presentation and also a beautiful poster, I must say. Yeah, I can start us off with the questions again, but for your future directions, you mentioned conducting more MST experiments. I'm not going to attempt to try and say the virus's name so I don't butcher it, but would you be following a same methodology for those experiments as well as you did for this one?
So something that Tyler had experimented and wrote previous papers on was that JEV as well as Zika interacted in solution with a human helicase dead box protein called DDX3X. So what we were going to look for was to see whether Murray Valley encephalitis virus and Powassan virus interacted with that same dead box helicase human host protein or not. Which is kind of like a not a side project, but just a little different from the cyclization sequence that we found in JEV.
That makes sense. Thank you. If there's any questions regarding Amber's presentation, please feel free to raise your hand and ask it, or you can leave it in the chat and I can also read it out. But this is great. I'm just admiring Amber's poster right now. I want to read out Trisha's comment because it's just so funny. It says, thanks Amber and Tyler for this work, and we can come back up and help Tyler finish the paper. So clearly you made a very good impression. Thank you, Amber. If you have any questions, we will have a lot of time towards the end too, so students will hang back so we can take questions then as well. But I will move on to our next presenter. Our next presenter is Kieran. And Kieran is representing LCI in the HYRS program this summer. He has spent his summer working with Dr. Boris Demler in his lab in the Department of Chemistry and Biochemistry. I will invite Kieran now to share his research with us.
Over the course of the summer, I had the chance of working alongside Amy Henriksen and Boris Demler on a project we decided to call Adeno-Associated Viral Characterization with Density Contrast AUC. So the title may seem a bit confusing right now. I hope it makes a bit more sense later. But before I get into the main portion of my research, I'd just like to touch upon the first portion of this title, the adeno-associated virus. So adeno-associated viruses, or AAV for short, are currently being used in gene therapy as means of transporting desired genetic information into patients. This then can be used in order to combat a bunch of possible and future viruses, such as genetic defects, diseases, and viruses. So this is specifically known as a viral vector, meaning that the adeno-associated virus is a virus. We are currently using a virus and their means of transport in order to carry the genetic information within their protein capsid or their protective shell into the patient. There is, however, a big problem with this current means of production and purification with AAVs, and that is the fact that we are not only producing loaded AAVs, which contain the genetic information we want, but we are also producing empty AAV capsids, which do not contain the genetic information we want and just cause an increase in immune response within the patient, making them seem more ill. So the goal of our study and the goal of our research was to use an analytical ultracentrifuge, AUC for short, and that is basically a spinning machine that spins really fast. In this case, we were able to spin it up to 60,000 RPM. And then it fires light wavelengths through our sample in order to identify certain properties such as sedimentation, diffusion, and other characterizations of the macromolecules. So using this AUC along with a density gradient forming cesium chloride solution, which I'll get to over in this part of the poster, we were hoping to be able to separate and identify the loaded AAV capsids from the empty AAV capsids, as well as other contaminants. This then can be used to determine the overall purity of our analyzed sample. So before I get into the cesium chloride density gradient, all you need to grasp from this figure 1 is that empty capsids have a lower density relative to the full capsids, which have a higher density. So what is the cesium chloride density gradient? Well, during centrifugation, we placed our AAVs within a cesium chloride solution. And then this cesium chloride solution, while it was undergoing centrifugation, started sedimenting, which means the heavy cesium ions started getting pushed to the outskirts of the channel being where we placed our sample. So this causes a high density region over in this region of the channel and a lower density region over near the center or this outskirts of the road or the channel. So why this is important is because it allowed us to exploit this difference in density within the empty and loaded capsids. As these AAVs started floating around and moving around within the channel, they started searching for a region in which they could reach their equal buoyancy, or in which their density would match the surrounding density of the solution. So because of the difference between the empty and loaded AAVs, the empty AAVs would find their equal buoyancy more near the center of the channel, whereas the loaded AAVs would find their equal buoyancy farther out in the channel in the higher density region. So what did we get as results? Well, we got this very nice graph here. What you see is two very defined peaks that I've highlighted as A and B. So this A peak occurs near the center of the channel and is smaller than the B peak, indicating that we have a small amount of empty AAVs. And then farther down in the higher density regions of the channel, kind of over here, we have a larger peak, indicating a large amount of loaded AAVs. Now, how we receive this graph is by using something called their absorbance properties. So how AUC works is it fires wavelengths through the channel, and the regions in which there is buffer or air, as you can see by this red or area called air, you see that most of the wavelengths will pass through that and hit the sensor. This then means that there is a low absorbance within the sample, creating a low line on this graph. That's why it's low compared to the peaks. But once we reach the AAVs, they begin to absorb the wavelengths and less hits the sensor, indicating that there's something there. And this is in turn shown as a peak. And then the more there are of AAVs, the more wavelengths are absorbed. And so the peak begins to grow, indicating whether there's more or less. That's how we know there's more loaded than empty. We then used a program called Ultrascan 3 in order to analyze the obtained data with absorbance and receive this graph that you now see in figure 3. At the same time as we ran this, we also ran an orthogonal validation method, meaning we ran a separate means of analyzing the AAV sample. In this case, we used a sedimentation velocity experiment. This means that we placed our sample within a low density buffer, and as it was spinning, we observed its sedimentation or its movement within the channel until it reached the bottom of the channel. We call that pelleting. We also use something called multi-wavelength analysis. So multi-wavelength analysis is when you fire a variety of wavelengths through the channel. And this is in order to exploit their spectral properties. What this means is different macromolecules, in this case DNA and protein, like to absorb different wavelengths. So DNA would appear more commonly at a certain wavelength, whereas protein would appear more at a different wavelength. We then were able to use this to separate the data pertaining to the DNA and the data pertaining to the protein to analyze them separately. We call this spectral deconvolution. So what you're looking at in figure 5 is a Van Holt-Weiss graph depicting the boundary fraction. This means the amount of the sample sedimenting at a specific sedimentation coefficient or speed. So what you're able to see is this pink line, which indicates protein, has about 70 to 80% of the sample sedimenting around the 95S or 95 sedimentation coefficient. You also have an equivalent amount of DNA indicated by the green line sedimenting at that same speed and at the same amount. What this indicates is that we have a large amount of DNA within the protein capsids, indicating loaded AAVs. We also have a small amount of proteins without DNA within them in this 40 to 60S, indicating some empty AAVs. And this correlates very well with the graph we received in figure 3. So as a conclusion, I was very fortunate because my experiment worked exactly how we intended it to. We were able to determine not only the contents of the sample, specifically the presence of loaded and empty AAV capsids, but we were also able to determine a rough amount of empty and loaded AAVs present. And for future works, we intend to calculate the V-bar of AAVs and determine the exact ratios between empty and loaded capsids. Before I open it up to questions, I'd just like to say thank you to Amy Henriksen and Boris de Meller for helping me out through this process. I learned a lot this summer and it was a great time. Thank you.
Thank you, Kieran. It was a great presentation, also a great poster. I have a question. I was just wondering, you will clearly know that I'm not a biochemistry major, but what exactly are like V-bar calculations, and I guess what the purpose of those are for your future directions?
Yeah, so simplistically said, the V-bar is the inverse of density. And this is useful because it allows us to calculate the molar mass of the AAVs, which we can then use to determine the amount of DNA and proteins, as well as DNA within the protein capsids, which is very useful for other researchers.
That was great. Thank you. Very good presentation. If there's any questions for Kieran, please feel free to leave them in the chat or raise your hand and ask them. But oh, there is a question. Could you distinguish partially filled versus fully filled capsids?
So that's a good question. We did some other runs and we saw some peaks show up that might have been partially filled, but we haven't been able to run a sample that we are confident enough to say there is partially filled within that sample. There is, however, a possibility that it would show up, and it most likely would. And where it would show up is in the middle region. Basically, it wouldn't be as dense as the loaded, but it wouldn't be as low of a density as the empty. And that is probably another run we would be testing later on.
Great answer, Kieran. Thank you. Yeah, if there aren't any questions, then I will move on to our next presenter. But like always, there will be a lot of time in the end for any questions. If you have any questions and you want to come back and ask the students as well. But with that being said, our next presenter is Finley Lothian. Finley is representing Catholic Central High School in the HYRS program, and she has spent her summer working with Dr. Uta Kote and her lab in the Department of Chemistry and Biochemistry. So I will invite Finley now to share her work with us.
All right, as Zainab had mentioned, I have got the incredible opportunity to work in the Cote lab this summer amongst incredible lab mates. I have learned so much from them. They're just amazing. And our project this summer focused on investigating the interaction of MNMG and tRNA glutamine UUC. So hopefully during this presentation, you'll learn a little bit more and I will not confuse you. So as we know, tRNAs are essential for protein translation and the survival of the cell itself. And before I get ahead of myself, tRNA stands for transcription RNA, sorry. And so in order to optimize tRNA functions, there are enzymes that are recruited to perform post-transcriptional modifications. In the E. coli cells, MNME and MNMG proteins are crucial for the smooth translation of proteins. So MNME and MNMG are homodimeric proteins, meaning they are composed of two protein monomers. So if we look down here in figure 1 and 2, you can see there are two protein monomers together, and they compose our MNMG and MNME proteins. So those are simple terms of how to explain that. And both come together and form our complex MNMEG, which these two are interdependent on each other, and they, I guess, are crucial for our MNMEG protein, which is very important in our body and in the E. coli cells. So you may wonder, why is it important that we're studying this? Why am I looking at these? So in humans, the MNME and MNMG homologs modify the U34 in a set of mitochondrial tRNAs. These defects affect mitochondrial translation, impair mitochondrial oxidation phosphorylation, and cause infantile hypertrophic cardiomyopathy with lactic acidosis. So in simple terms, this just means that it affects infant hearts. So it is very important we look at this. In this study, we tested the interaction between MNMG and tRNA glutamine UUC. To do this, we expressed and purified the MNMG E. coli complex and analyzed it with size exclusion chromatography. So size exclusion chromatography is a system where we separate molecules by size as they pass through a column. Large molecules come through the column faster than the smaller molecules, thereby they are able to separate the molecules by size. The tRNA glutamine UUC DNA template will be amplified from a plasmid prior to transcription. After separate analysis in this SEC, MNMG and tRNA glutamine UUC will be incubated together and analyzed to see if there is any interaction that occurs. In the first step of our experiment, it was to purify our MNMG and MNME proteins. The purification was performed using a nickel affinity chromatography by gravity flow at 4 degrees Celsius to avoid any potential protein loss. Then, to finalize the purification, MNMG and MNME was subjected to size exclusion chromatography. Well, unfortunately, I learned the hard way on what scientific research is really like, and our MNME was lost in the SEC. It did not show any UV reading, which means our protein was not present in the fractions that came off. Although our MNMG fortunately showed on our UV readings and provided us with a nice UV curve as shown on figure 5. So then we decided to move on to the next step after we successfully purified MNMG. We moved on to the PCR of our tRNA glutamine UUC. PCR stands for the polymerase chain reaction, and this was performed to amplify our tRNA glutamine gene that will be used. We repeated the cycle of denaturing template and synthesizing DNA 25 times. Primers were annealed to the template at different temperatures, which all produced the tRNA glutamine UUC gene. After our PCR, we took all of our tRNA glutamine genes that were produced and performed an in vitro transcription. The IVT replicated the process of RNA synthesis outside of the cellular environment. Thereby, our tRNA glutamine was synthesized in the experiment. And if we look at all of the bands in both our PCR and our in vitro transcription, we can see our ladder that is produced, and it all lines up. And here we can see it is present in our urea page. So we got the go-to the next step. Upon both MNMG and tRNA glutamine being purified and synthesized, we decided we were ready for the sec run. We decided to run MNMG alone, tRNA glutamine UUC alone, then finally both together. We decided to use this method just so that we can make sure our protein and our tRNA was there, and we can see which fractions they come off in, because that is very important for determining if they do interact or not. Our MNMG sex fractions kind of showed a little bit disappointing, but showed there was very little protein, if there was any protein present. As you can see, the lines are not present for the rest of it. You can see our protein ladder and pre-SEC run in this column, and you can see where our protein is supposed to be and the blank lines that exist. But despite that, we moved on to our tRNA UUC sec fractions. And this was a very exciting one. So during our sec fractions, our tRNA glutamine UUC, the UV light showed it came off. And in our fractions, about 30 to 40, we can see beautiful dark lines in our urea page. So this means that it's present. So we decided to move on and put them both in the column. And as expected, with our lack of protein due to precipitation, the fraction showed that it came off, the tRNA came off in the exact same fractions, which means it did not interact. It just fell straight through. So to conclude our experiment, we were able to successfully purify MNMG E. coli proteins. Our tRNA glutamine UUC was transcribed and purified. We saw the tRNA glutamine UUC was stable during SEC analysis and provides adequate absorbent profiles for complex formation determination. As for future directions, we may decide to include MNME, as it may increase the solubility of MNMG in the solution and improve binding to tRNA. Additionally, creating a more stable buffer for the MNMG protein to be in, as MNMG can be more soluble and there's less risk of precipitation or protein loss, which is a problem we did encounter. And I would like to finish it just by acknowledging Uda Kote and the lab members and how supportive they've been and how much I've learned in my experience with the HYRS program.
Thank you, Finley. That was an amazing presentation. We will take Finley's questions now, and I'll start it off. You did mention a little bit about a buffer. Do you have any suggestions on what buffers could be used for future experiments for the, again, I'm not going to try and mention the protein name so I don't mess it up.
Yeah, to create a more suitable buffer for the MNMG protein, we could possibly add glycerol, a detergent, a reducing agent, or salts, as it would ideally help MNMG and stability in solution.
Makes sense. Thank you, Finley. Any questions for Finley? We will be happy to take them right now. But I do have another question, so I might as well go. You talked a little bit in your presentation about a UV curve, and I was trying to figure out what a UV curve was. So do you mind telling us a little bit about what that is?
So for our SEC fractions, one of the easiest ways to kind of determine if our protein or tRNA or whatever you're dealing with is present is looking at a UV curve. And a UV curve is a change in ultraviolet absorbance, which is detected by our SEC machine. So they can determine it and kind of have a good idea on what's present, how much is present, and then we can kind of see what fractions it should come off in, and we can see which fractions we should collect. So if you can see on my screen here, our MNMG SuperDex purification, it just looked beautiful. So we actually collected these fractions and used this purification fractions for our next run. So we just looked at the UV, we looked at which fractions it came off in, and we collected those and were able to use them for our next step in our experiment.
Beautiful. That makes sense. Thank you. You're doing such a great job at explaining things. It makes a lot of sense. Thank you, Finley. It was a fabulous presentation. I will move on now to our last and final presenter, but we do have we are quite ahead of schedule, which is always amazing. So we will have a lot of time in the end if you want to just interact with the higher students and ask them any questions. But our next presenter is Zaki Olvis. Zaki is also representing Catholic Central High School in the HYRS program. Throughout the program, Zaki has worked with Dr. Stacy Wetmore and her team in the Department of Chemistry and Biochemistry. So I would like to invite Zaki now to share his research with us.
Hi, my name is Zaki Olvis, and I'm going to be presenting about the nucleobase and sequence-dependent'properties of 2' O-methyl modified RNA. And just like insight on what I'm going to be presenting about, it's just that we're going to be studying how chemical modifications on the RNA will affect the structure of the duplex. And I'm going to explain that. So RNA therapeutics right now is a really hot topic, especially with the COVID-19 vaccine that uses mRNA technology. Our study focuses on small interfering RNA, or sRNA, which is a double-stranded RNA that's involved with mRNA regulations, and it offers a lot of therapeutic capabilities. But unfortunately, the thing is, RNA by itself is a really poor drug, just because there's a lot of mechanisms within our body that prevent the uptake of RNA into the cells, meaning that foreign RNA can't go inside the cells. And to be able to overcome these pharmacological obstacles, chemical modifications have shown various successes, especially at the 2' position. So I'm actually gonna zoom in here for my example. So one of the prime examples of modifications overcoming pharmacological obstacles is Givlaari. Givlaari is a FDA-approved siRNA-based drug. And as shown in the figure, the entire duplex of Givlaari is modified at the 2' position, which is like here. And what modification at the 2' position mean is just this. So if you look at the 2' position, which is highlighted in red, there is this hydroxide. There is this hydroxide group present, and we're just substituting that with an O-methyl. And what makes this position really interesting is if you look at this figure right here, here on the left side is a DNA. And as you can see that when we add a hydroxide, it becomes RNA-like. And so it just makes it more compact and already has a structural effect. And if we look at more on the position of the 2' or hydroxide, here in the bottom figure, you can see that on the red, there's close proximity to these green and yellow blobs. These green and yellow blobs are just hydrogen donors and acceptors within what we call the minor groove solvation networks. It's just an extremely important water system that helps with the stability and structure of the duplex. And so what my research is just about is using computational chemistry to understand the effects of these modifications at the 2' carbon position. So moving on to the computational details of my project, we actually use molecular dynamics simulation, which is an all-atom simulation that represents the atoms as ball and springs. And our simulation blocks are very small, containing a solute, which is the modified RNA that's surrounded by waters and ions. And we simulate this at a given temperature for 500 nanoseconds. And moving on here, these are just the 4 duplexes that I'm going to be showcasing today. And we have 4 different duplexes as we take account not only for the nucleobase identity, but also its neighbor. So that's why we have here, it's just like an all adenine strand, and we also have an alternating strand. And all of these strands actually have 18 base pairs, yeah. And we actually use a modification system that uses 3 modifications on the strand that's evenly spaced so that we can optimize the strand basically. And these parameters are actually just the movements of the helix, but we're going to be looking closely more at the base pair parameters. And what does this mean is that base pair parameters are just looking at the basic movements of like the nucleobases with regard to each other. For example, this is a base pair right now, and a buckle movement would be like this, and a propeller action would be like this, and an opening motion would be like this. And so that's just kind of like the base pairs that we're looking at. And to be able to visualize the difference in the base pair parameters, we actually compared the modified structure with an unmodified structure. And here on the y-axis, you'll just see here like the buckle, propeller, and opening that I was talking about. But here on the bottom are actually the base pairs that we're going to be examining. So it just goes from residue 1 and 36 to 18 and 19. And here is just the legend where just the red parts are just meaning that the base pairs are distorting in a positive direction, while the blue ones is distorting in a negative directions, and the white is just like a neutral or like 0 deviation. And if you actually look at all of this at once, you'll actually see that there's a general trend. Oh wait, sorry. And we actually look at like the yellow boxes here. That's actually the modified strands that we're looking at. So here is the modified strands and the modified bases that we're looking at. And around it, we can see like a blue box, and this blue box are just its neighbors. And we can actually see that the modification on this base is actually affecting its neighbors. So if we actually look at all these fingerprint regions, which are just these blue box, we'll actually see a very general trend in how variations and how modifications affect the modified base and its surroundings. However, we do see that there's variation in each. However, like there's variations in existing with regards to the nucleobase identity and the sequence context. However, this small subtle variation can make a huge difference when in systems where modifications are heavily applied, like here in Givlari, where the entire system is all modified. And moving on to the minor groove solvation disruption, the radial distribution function just tells us how much water is present at a certain distance relative to a particle. And so for example, here on the C2' oxygen, we are looking at this oxygen right here. And we are just looking how much waters are close to it or how far it is. And so when we actually examine the graphs itself, we can see that on the first peak here, that all of it is kind of like uniform. So they're hydrogen bonding at these peaks. However, when you look at the secondary solvation, which is around here, you can actually see that there's a difference between the modified and unmodified, with the unmodified being the green and black strands, and the modified being the red and blue strands, where the red and blue strands are actually farther away than the unmodified counterpart. What this just means is that the methyl group is actually displacing the water away from the C2' oxygen. And if we look at a radial distribution graph relative to the minor groove acceptor, which are just these yellows here, we can actually see some very interesting phenomena. And we can observe that the impacts of the modification, that we see behaviors that are both nucleobase dependent and sequence dependent. And looking at this guanine modified duplex, you can see that the solvation is actually really disrupted. And when sequence context is taken in, you can see that the green here is actually a unmodified poly-CG. It just means that like, depending on the sequence context, the intensity of this disruption is varied. But one of the most interesting behaviors that we have ever seen is right here, is in the uracil modified. The reason why it's so interesting, because if you look at it, there's almost no impact on the modification. There's no impact on the minor groove solvation networks by the modification, basically. And what it just means is that the modifications are not affecting, is that the uracil modified minor groove water networks remains unfazed or resistant to the effects of modification, leading us to believe that the uracil is a prime candidate for the modifications when we want our minor groove salvation intact. And so overall, the structural parameters are nucleobase and sequence dependent. And although there is a lot of similarities in behavior, there's also a lot of variations occurring. And when it comes to the minor groove salvation network, we see that the effects are heavily dependent on the nucleobase and sequence context. Overall, this study just provides a foundation on the sequence and unique nucleobase effects of the 2' carbon modifications, considered part of the rulebook that would contribute to our rational design of future RNA therapeutics. And before I open for questions, I would just like to thank the HYRS program for giving me this opportunity, and a special thanks to Dr. Stacey Wetmore for making me part of her lab.
Thank you, Zaki. What a beautiful presentation. First of all, Zaki's poster, everybody. That was a wonderful poster. Thank you. I do have a question. Not a very detailed question, but what would be the future directions of your research here?
I mean, like, as I said, like, this research just provide like a foundation on the rational design of future RNA therapeutics. But in terms of research, this is still very ground tradition and very basic. So there's a lot of things that we don't know yet. For example, is in the uracil modified. So like we know like uracil is resistant to the impacts of these modifications, but we actually don't know what makes uracil unique that it makes that behavior, basically.
Makes sense. That makes sense. Thank you, Zaki. It was a great presentation. Wonderful, wonderful job, everyone. Thank you. I think we are quite ahead of schedule, but that is amazing. Thank you all for joining us today and celebrating the research and hard work of all of these students. Thank you again to Alberta Innovates, the University of Lethbridge, all of the primary investigators, but also all of the lab supervisors. You have all been such great help and have really mentored all these students and helped them learn and grow. Again, thank you so much to Valerie Archibald and Catherine Reeder as well for all of their support. I would also really like to thank all the local high schools that have always been involved in the HYRS program. We always appreciate your support as well. With that, we will end the event like all the